Okay. All right. So many of you all are living the physician scientist dream, but I wanted to kind of give you some background into how I came to this profession, and sort of how, because some of us also are thinking about how does your career goals evolve over time as well. So not only how do we get into it, but then how do you maintain feeling like you're really contributing in as many ways as you're able to, as you sort of get up in the career, that get more advanced in your career, that ways that you're able to do that you weren't previously. So, so one thing that I typically try to impress upon trainees, and leading a National Pediatric Scientist K-12 program, I get to do this a lot, is that the training period should not be seen as finite, and really seems long in the medical world, but it's not that long when you consider the rest of your career. So, we spend, if you're an MD, PhD, 7 to 8 years in medical school or grad school, at least 4 years in medical school, 3 to 5 years in residency, 3 years in fellowship. But all that time added up doesn't come close to even half of the number of years that you'll be working in your selected field. And so really the training should be just seen as part of the career, rather than it's a bimodal framework. And so most of this length of training and then career should be, not only how do you prepare yourself for the next step in your career, but also maintain that productive and satisfying career, because it's going to change and evolve over time. So just to frame a little bit for you how my career path played out, and Mary talked about that a little bit, but I was actually a PhD before I was an MD. And I'll kind of tell you the story how that evolved. But so I took an unusual path to begin with, that I was in my first year of my PhD and met physician scientists who were going back between the lab and the clinic, really studying RSV. They were peds ID docs. And I thought, oh, wait, I want to do that. And so I applied to medical school while in my PhD, and hadn't even taken the MCAT. So I had to study for that during my PhD. I then decided to train in pediatrics, did pediatric infectious disease. I had my family, got married during residency, had children during my infectious disease fellowship and early faculty years. And then was recruited to an assistant professor position at Duke. And this is one that I called them actually to say, do you have a position for me? Would you have a startup package for me? And that is, not totally uncommon, and something that I was glad to be able to be in a position to do that, because I actually lost my sister, who was 39 at the time, and she had 3 kids, and I wanted to move to where her family was and be there while her kids were getting through the rest of elementary school. So that was where I had my first assistant professor position. I did start a number of projects that I continue to work on today while at Duke. I became an associate dean of physician scientist development, because I kind of got interested in, okay, I think I know how to do this physician scientist thing. How can I help more people do this? And then I got a call about considering a department chair job, something I never imagined I would do, starting back in even just in early years at Duke. But saw that I'd seen the opportunity to change the way we pursue research, pediatric care, and thought that could be a next exciting opportunity for me, and continues to be. Here's some pictures of my progeny and my husband there. That's at now we go to Yankee Stadium to see the Red Sox. So talk about, kind of a formula for setting yourself up for a successful physician scientist career, or something, a talk that I've sort of evolved over time, and gonna give you a little insight into what I thought were the most important pieces of physician scientist career, and now how it's extending into not only the research that you do, but what else you add into your career. So first and foremost is, and this again is in the framework of things that you can pass on to your mentees as well, is you got to discover your passion. If you're not passionate about what you do and get up and do every day, because none of our pieces of our jobs are easy, is you have to feel like you're doing good for the world and that you care a lot about it. So I came upon this early on when I was in undergrad, and I was from North Carolina. I had really not left the state so often, definitely hadn't left the country. And I just, when I got to college, there were all these opportunities to go different places in the world. And I thought, like, I need to get out of here. I need to experience like something else. And so an opportunity that sounded of interest to me was to go to a small rural hospital in a rural part of Zambia. It was the mid 1990s, and so HIV pandemic was the epicenter was in sub-Saharan Africa, and there was no therapy. So I traveled all this way, my first time out of the country, and arrived at the town of Mwandi that looks about like that top picture. It was clear that this tiny virus was causing havoc on this society to me from day one. There were children everywhere. They were being taken care of by their grandparents because, usually their grandmother, because their parents were in the hospital dying of TB or some other opportunistic infection. And that was the status of the town. And then, even to add more insult to that was there were children dying as well, young babies that were dying of the same virus as well. And the only thing you could say is abstain, be faithful, or condomize, as the sign says, or die. I mean, that was essentially the status. And I thought, wow, like, how does this part of the world ever change this trajectory? And it sort of like hit me like a lightning bolt, like, well, a vaccine, they need a vaccine for this. Of course, I didn't realize what a challenge that was going to be back then, but that was kind of where I got my start in. A vaccine would change the trajectory for a place like this. So what I tell trainees is, you got to make finding your passion your job. You need to know what motivates you. And in order to, be successful and have a career in this as a physician scientist, it has to be an important question to human health that is fundable by funding agencies. What unique skill or aspect might you bring to the field? So I think where innovation happens, it happens when you apply a new set of thinking or expertise to an old problem or to an old field, or a field that's been sort of going on one trajectory and hasn't had a new set of innovations applied to it. So that's always a nice niche to find. And of course, is it on the strategic agenda of the NIH? So no matter how many other funders there are, NIH is still our workhorse, and so it has to be a priority there. And if you haven't found your passion, make it your job to find it. And then that's kind of where you can start, right? And then I also promote that you got to keep getting the experiences that are going to motivate you, because I think one advantage we have as physician scientists is we, discover our passion, whatever the disease is, whatever the patient population is, the metabolic pathway, whatever it may be. And then we get to experience people whose lives are affected by that. And that's a really unique thing that, PhD scientists honestly don't have the same opportunity to do. And so you need to put yourself in places where you continue to get that motivation, because I think that really is a key to success of, all the hard things that we do every day. The reason is because we want to make a difference for that patient population. So, my opportunity was to continuing gaining experiences in areas of the world that were being affected by HIV. So I went to other settings for in my training for healthcare in Zambia and as well as Malawi. I even sent, when I was a assistant professor at Duke, one of my medical school student trainees who was in my lab, she went to the same mission hospital where I had worked through my connection. And that was great to be able to send sort of the next generation to a teaching hospital that was led by a missionary physician who had taken on malaria research as part of his work. And so, I continued to do that on repeat for a little bit while I was sort of gaining, okay, what area of research? I've gotten my motivation. I'm getting my experiences that continue to make me motivated to, protect mothers and infants against HIV. And I then started integrating research on top of that. So in addition to doing as many residency and fellowship rotations that I could get out of, the hospital itself, to get credit for doing them in a global setting, but also started to integrate research. Well, could I enroll a population while I'm there? So that was sort of my way in of getting the experience, but then also going towards what a career, what a potential solution that I may study. But of course, along the way, you realize you didn't get all the training in medical school or even in your PhD, that you are going to need to apply whatever scientific expertise that you're going to apply to then make new solutions for diseases. And so, for me, it was, again, starting as a PhD scientist, where I did choose a PhD department that was focused on public health, and the PIs in that department often focused on a specific disease rather than a basic immunology department where you may focus more on an immune cell population or signaling pathways. And this is where I ran into infectious disease specialists who I just thought that was the coolest thing in the world, that they would go see a baby with RSV and then they were studying RSV. Of course, we're still studying RSV, especially right now. And so I thought, well, I need an MD, and went to my mentor, because your mentors are key in all of this when you start, and said, what do I do? I think now I want to go to medical school. Luckily, she said, I will help you. I'm an MD PhD, and it was the key to all of my work now. So, so if you've determined that you need the training or the course, like, implement it right away. And I think this doesn't apply just to when we were, getting our initial graduate degrees. It even applies now. And something I still, try to sneak in when I can, like, let me learn more about the single cell genomics approaches. Let me learn R. These things that we don't need to not pursue new trainings now, even into our career, though we may lean more towards sending mentees to those types of trainings. But if you identify that it's a need, like, get the training course, and luckily we still work within academic settings in which often training is available. So then along the way, I think we probably all experienced that many people have opinions on ways your career will go, and things that you can or can't pursue as part of your passion and your questions that you're asking. And one, I was just talking to my department chair friends about this. One thing that I can characterize about myself, and my husband would say, would be fully on board with this, is like, I don't usually accept no for an answer. Like, I might eventually, but not initially. And I think there were many times when I heard no for an answer, and it honestly just made me a little more motivated. So like when I was a PhD student and I was applying to medical school, I heard a lot like, well, you can't do both of those at the same time. You'd have to go into a joint program. And well, you couldn't do an MD at another location if you were doing your PhD at a separate location. I ended up applying to schools all over. I thought, like, it was this horrible thing that had happened to me, that I did not get into medical school where I was pursuing my PhD. I did not get into Johns Hopkins where I was pursuing my PhD. I thought that was a big blow, but it actually turned out to be the best thing, because at that time, the Hopkins MD program was still that, go to class 8 to 5, you memorize everything, you take a test on everything. I got into Harvard, which, I'd only applied to because I was applying to Hopkins, really. There they had changed over to the new model, the new pathway of you go to class like half the day, and the rest of the day you're supposed to Google stuff. You don't memorize anything. You get together with your study group. And I just used that opportunity to go to the lab, basically. And so I got a lot more done being able to go to a medical school that was not at the same location as my PhD program, and initiated a new mentorship in that program. So, and I remember even getting called into the registrar's office like halfway into my first year, and they said, oh, we see you're still enrolled at Johns Hopkins. You can't be enrolled in 2 different medical schools. And I said, well, when I applied, that's what I said I was going to do. And you admitted me. So I'm going to keep doing that. And somehow the registrar just accepted that. So, so don't take no for an answer. I heard real scientists go into medicine, not pediatrics. Hopefully we don't hear that anymore, but honestly, my like favorite mentor of all time told me that, and he was serious. But you know, I didn't accept it. My thinking was that, you know, I want to develop vaccines that give people lifelong immunity. The best time to start that is in pediatrics. We deliver most of our vaccines during early life. I don't see why I wouldn't go into pediatrics. And so, that was maybe the one time I didn't take his advice. One of my initial areas of study was studying lymphocyte populations in breast milk. I heard a lot like, oh, there's not enough cells there. You would never be able to study that. It just made me more motivated. I did it anyways. This is maybe one that's really, really unique to certain non-human primate researchers, but I thought, oh, I really want to make non-pregnant monkeys lactate so that I can study breast milk more easily in the non-human primate model, and I wouldn't have to wait for them to be pregnant and all of that. And everybody's like, that's nuts. You won't be able to do that. Well, I just got more motivated, and I, you know, read papers about how adoptive mothers breastfeed, like can do non-nutritive breastfeeding, and I applied that to monkeys, and we got them to lactate, and so that's a strategy that others have used now as well. I'll tell you a story about how I got into cytomegalovirus research coming from the HIV world. I was really motivated by just my clinical practice and seeing how many kids are affected with congenital CMV. I wanted to make a non-human primate model because that's what I was used to using in the HIV world. And everybody said, oh yeah, I wouldn't bother. Like the non-human primate CMV viruses don't cross the placenta because we've never seen, you know, a case of congenital CMV in monkeys. And I thought like, I'm not sure that that's true. And so I want to test it experimentally. So I did that. And something I hear a lot now is working on developing an HIV vaccine that you would deliver in infancy for lifelong protection. And a lot of people say, well, you can't enroll children or infants in a vaccine trial if you don't have efficacy in adults. Well, I think we're changing that paradigm, and we saw how far that put us back in the vaccine development for COVID for children. And so, so I'm working at the policy level to change that. No. So just, you know, don't take no for an answer, prioritize your goals and go for it. Is my my MO. Sometimes you do have to accept no, but you know, push it first. I think we all know at this point how important your mentors are and career advice that you can get from them. But, you know, it's on us to really use those mentors and continue to use mentors throughout our life. You know, they don't tap you on the shoulder all the time and say, let me give you this good advice. You really have to seek it out from them. So I started my career with this wonderful set of mentors. Diane Griffin was my PhD mentor. She was the one who was not fazed when I said, I'm a PhD student. I have not even taken the MCAT, and all of a sudden I've decided I want to go to medical school. She said, great, let's make it happen. And even when I went to her and I said, I think it's gonna be too hard. Maybe I should just get a master's degree here and go to med school in a different city. And she said, no, you're halfway through your PhD, you really shouldn't do that. So I have to hand it to her that I did stick with getting the dual degrees. And she is a remarkable woman who, again, was an early MD PhD virologist and neurologist, probably the only person that has grants in measles virus at this point, that still needs probably a new vaccine to really eliminate. And she is stepped down as department chair, but she's still an R01 funded faculty member at Johns Hopkins, definitely well into her 80s. And we have shared now a lot of mentees where she sends me some of her graduates to be postdocs, and we've had success doing that. Norm Leffen was my mentor during my MD program. He was one of the discoverers of SIV and the non-human primate models at Bates. He was like a second father to me, and honestly, he was like 3 standard deviations above the typical mentor in terms of things that he did for me, and really how much he advanced my career, so that every other mentor looks, not adequate compared to what Norm gave me. So I did have to reset my expectations. And he unfortunately passed away at an early age, and that was hard. I went through a process where, the person who I thought mattered most to me for my career had a terminal illness diagnosed, and so that was a little bit of a wake-up call for me, and maybe is part of the reason I looked to get independent so early, because I saw how critical that was. Because at the time he had his disease diagnosed, I thought, well, that's horrible for him, but it's horrible for me. And then I had so much guilt that I thought that as well. So I learned a lot through that process. Dr. Bart Haynes was good friends with Norm Lettvin. He was the one who helped me get a position at Duke when I had a family tragedy. And that I do a lot of things today because of watching Bart and lead big programs in vaccine development. He was also a clinical department chair prior to his role. So these three were really critical in my career. And I think, all of us can probably mention at least three, but probably more. And I continue to have mentors today, and also call these individuals back as well. And Norm, I just remember some of his really one-liner advice that he was so good at that comes into my head every once in a while. So who makes a good mentor? In your selected area of passion or somewhere related, pushes you in your career, but important for women, I think, is not threatened by your success. Definitely I have experienced mentors that were threatened by my success, and that was confusing to me, because I thought, why would they not be so happy for my success? And you have to understand that human nature is competitive, and there is that mentee succeeding their mentor issue that arises probably in the majority of cases, and you have to be ready for it. If it hasn't happened to you in your career, it likely will. I was not prepared for it at the time it happened. I remember it was my mother who said to me, Sallie, I think you're being bullied by your mentor. And I said, no, that can't be it. And then I started talking to a mentor, a peer mentor this was at Duke. And he's like, yeah, that's definitely what's happening. And it was my mother who had to point it out to me. So be aware that these can be blind sides. They need to network for you and for others, so that they, one way to screen for that is to look for someone who has done that for others. One thing that I tell mentees when they're looking at mentors, screening them, is just look at the publication records that's publicly available for those individuals. Look, have they had first author papers with that mentor, or has trainees and junior faculty had first author papers with that mentor, and then you see it swap. Has that happened for that person? That's a good sign for someone that is interested in their mentees advancing and even beyond their own successes. You will definitely need more than one, and I think that's hopefully something that all of you have already come to, and that continues to change what your mentorship needs are. One thing that Norm Lettvin did for me, and now I realize how unique it was, is when I was at the end of fellowship and trying to decide what would be my junior faculty research area. Should I stay in HIV research? Should I go to respiratory viruses? What should I do? Something in vaccinology in children. That was sort of what I had in mind, but which virus? He said, okay, Sallie, let's go around and meet all the great virologists at Harvard Medical School and ask them, what if they were you at this moment, what would they study? And so we did that. We went to 3 or 4 virologists across the university, sat down for a half hour, and just talked about that. That was something that, honestly, I haven't ever heard of that since. And I can't even say that I do that for people now, to spend, that much time helping someone, think about their next phase of their career. But it's something you can also do on your own. People love to talk about themselves. And so just go talk to people about, if you were me now, what would you do? How did you become passionate about what you do? What unique training and experience did you seek? And how do you see that evolving now? Again, ask them the question, if you were starting your career today, what would you pursue? Okay, but all of that is kind of you're getting yourself ready for a career as a physician scientist. You're putting all your mentorship in place. You've got your passion project area. You've got your skill set. And then there's that moment, and some of you may be in this, where you got to focus. Like, you got to write a K award on something. You got to write your first grant on something. And it can't be 5 things, right? It needs to be like one thing. You know, you need to have that big manuscript that then establishes that you're in that field. So focus is a really critical part of that early career. I see it as kind of like a hourglass. Maybe the physician scientist career is, you start out kind of thinking about all the things that might interest you, and then you come to a time where, like, no, you got to really have one area that you are advancing. Yeah, I sometimes refer to it like the jig is up, like, enough experiencing everything, it's time to, you know, focus. It's got to be, you know, area of your passion, your skill set, something your mentor does at that point, because you don't have the funding to do it. So it has to be something that's related to your mentor work, that your mentor sees as an advantage to what they're already doing, and they can pay for it. And it's in an area that you've been productive already. Often I say, you know, 2 interrelated projects at the time you're like writing your first set of grants is often good, because you can kind of play them off each other. Like, okay, this grant didn't get funded by Burroughs Wellcome, but I'm going to submit it as an R21, and then I'm going to be submitting this other grant as a K award. You know, there's some things that you can benefit from having kind of 2 major hypotheses, set of aims that you're submitting at the same time, but they need to be interrelated, because again, you're focused. The pursuit has to be feasible. Again, I talked about this, the mentor working on it, practical, with, you know, priority for human health and at the NIH level. And there are achievable goals, those short-term and long-term goals. When I chose my focus, my passions were babies, viruses, immune responses, vaccines, developing world challenges. My skills were virology, immunology, pediatrics, and being a clinician. And my mentor was working on HIV vaccines and non-human primate models of HIV. So when we went around to all those you know, famous virologists at Harvard Medical School, a lot of them said, you know, you should really work on respiratory diseases. That's what's really going to kill people, you know, one day when the new epidemic comes, right? They didn't say coronaviruses, but they said flu. And so I thought, like, okay, maybe I should be a flu virologist. I previously studied measles. And then my mentor and I sat down, and he said, I can't mentor you if you want to do flu research. What I do is HIV. And so I thought, oh, that's a bummer. Like, I really like you as a mentor. And so I went home and thought about it, and I said, came back to him and I said, you know what if I take the focus of maternal infant HIV transmission and vaccine development there, and then you can continue to mentor me and I can do something that I'm passionate about. And we said, deal. So that, so working to develop non-human primate models to study perinatal HIV transmission was kind of the first area that that he said, okay, that's yours. I'm not touching that. So you can take that with you. I do very much promote that you should not stop investigating during clinical work. And I think that applies not only in training, but even ongoing in our careers. So, you know, I promote that it's just kind of unbelievable what we expect of people, right? When you come out of medical school, maybe you've done some research, you go to 3 years of residency, then maybe a year of fellowship, and then you have 2 years that's kind of like your postdoc if you want to be a researcher, and you're doing other stuff at the same time, likely, you know, 20% or so clinical work. And then we expect people to be ready for an independent career at the end of that. Well, it's kind of unheard of if you like compare it to a postdoc that is solidly 4 or 5 years of only focusing on research. And so, and it's hard to get that first job then and have that set aside academic time, and because academic medicine funds are not as flexible as they once were. And so, so this is, I think, problematic how we do it. So I think that you need to start in residency, pursuing research endeavors, and then you have a longer runway. So I tell medical students and that when you're looking for a physician scientist career and your clinical training portion of that, to use your residency interviews as really research potential mentors, and look for those residency programs that integrate research into their starting in residency. And that I see this is the future of making the physician scientist pathway more obtainable for more people. There's starting to be some funding opportunities and established programs. I know UCSF is well known for them. We've integrated one here at Weill Cornell. We did it at Duke too. So being able to initiate research that you may then carry on as your independent research throughout fellowship and then into faculty, I think is the way that more people can make it across the divide of becoming an independent scientist. And so you're ready for a career award by the end of fellowship, really. And then you're a sure bet for that department chair to fund. And even if they have to fund you for a little while waiting for your career award, it's a sure thing. In now becoming a department chair, I kind of see people that maybe they're from their K to R transition, or maybe they only came into the idea that research would be part of their career during their fellowship and therefore didn't get a lot of training time, or maybe they pursued one type of research and now are pursuing a different type of research. We've made a more standard process that was never kind of in place before, where you could apply for gap funds and make a 1 to 2 year plan of like, here's going to be the way that I go about setting aside research, pursuing a portfolio, and then getting it externally funded, and that we then evaluate those annually. It often in department it ends up being a kind of individual deal that you make with a division chief or department chair. And I think if you are proactive and say, okay, I want to go in this other direction, or I haven't yet made it towards getting my independent funding, but here's how long it's going to take me to do it, and here's the steps I'm going to do, I think you're in a lot better position to ask for funding, whether it be your department, whether it be your the dean's office, whether it be applying for a internal or foundation grant. If you can write it down what your external funding plan is going to be and how you're going to get there, people are a lot more ready to buy into it. And then, of course, the reality of to be a physician scientist, you have to be ready to apply for a lot of grants. Grants are not as easy to get as they once were back in the 90s. This is on the left is a graph by percentile score along the bottom. So if you got a 20% percentile score, almost all of those got funded back in the in the 90s. And now only about 25% of those that got a score of a 20th percentile will get funded. The age at which people are getting their first R1 is continuing to rise. This was only until 2014, but I have not found another graph that looks better. And then the age of investigators is increasing. So the way you deal with that is you just apply. You'll never get a grant you don't apply for. When you're starting out, grants, big or small, are an important measure of success. And I still apply for $50,000 grants frequently, because that's how you initiate a new project or you make a new collaboration. So I still seek out internal grants, foundation grants, instead of just, the bigger grants. Don't wait to be asked to apply. Seek all the grant writing instruction that you can when you're early on to this, and always ask people to read it before submission. I try to do this. I don't always make it before the submission to give it to someone to read. And now I work with a program manager who she reads it after I finish writing it, because sometimes it just doesn't make sense. And it's even better if they don't know the field well to review it for clarity. So that's how you become and stay an independent investigator. So then there's the survival part, because how do you write all those grants, see all those patients, actually pursue the experiments, mentor your own set of trainees, is first believe in your abilities to succeed. That's critical. You got to be persistent. Again, don't take no for an answer. That includes with grants. So I'm just getting an R01 now that I think it was my 4th submission, might have been 5th submission. Like if I believe in it, I just keep submitting it until it gets funded, because eventually you'll hit the lottery with the reviewers and get it funded. Sometimes I drop it because, okay, maybe that wasn't a good idea after all. But be persistent and especially if you're getting like decent feedback on it. But of course, be responsive to feedback. But so important is the importance of a supportive home life. So, establishing the expectations in your home life is really important, because it takes a lot of work, and it can be all minutes of your day could be consumed with being a physician scientist and pursuing your area of passion and pushing things through, and the amount of paperwork we have to deal with, and just the amount of demands on our time, that you have to have that supportive home life, but also be the person who also recognizes that there's a limit. Okay, I just can't do any more work tonight. I just can't go on that one more trip. Your partner's career will definitely impact yours. I am lucky in that Matt, my husband, knew that going in. I remember my parents, because I'm from the South, I guess Matt did ask my parents for my hand in marriage, which is definitely, not required, but he felt that was a good idea. And they said, sure. However, Sallie's really into her career, and we want to make sure you understand that before we agree here. And he's like, oh yeah, I know. And so that is something that he sort of said, like, I know you're into your career, and I'm going to be into that too. He also has his own career. He's a senior executive for communications and media at the Greater New York YMCA, which has 24 Ys across the metro area, that now he's responsible for their communications and marketing and branding. But he has committed to a career that says, I basically come home at 5 o'clock and I will deal with whatever the kids have after that time. I became the morning parent somewhere along the way. I think it's because, I was better at early rising than Matt was. And so I try to not do things before 8:30 in the morning, even as department chair. Now I have to break that rule fairly often, but I try to say no to things before that, because I'm the morning parent. I get everybody to school, then I clean up the house, put the dog away, and then go to work. And Matt's like out the door, he reads the paper, like he doesn't have kids in the morning. So that's how we've done things. And but you have to have that expectation setting, I think, before going into this, even before you put some progeny in the mix. Okay, so now kind of thinking about when you're out of the initial stages of becoming that independent investigator, is once you're focused and funded, it's really important that you take diversions. So this is the hourglass. So now you were focused, you got through the gauntlet, and now you need to not just do what you wrote in your aims. And this is where, you should have some resources. If you became an independent funded investigator, there should be some resources of funds that are available to you internally that you could focus on something other than what you wrote in your aims. And those diversions are really important, because they're where the innovation is going to come. That's where new opportunities for next phases of your career are going to come from as well. So I'm going to talk about just a couple of areas that I took diversions and it paid off, and some things that have long tails to them, some things that were one-off fun projects. So I'll talk about a couple of scenarios, but maybe before I launch into that, I'll just take a breath and see if there's any questions that anybody has so far.
I have a question. This has been really fantastic. So I wanted to talk a little bit. You kind of had this great story about how you called up Duke and said, hey, please, fund me. And I think that's fantastic. But, I wanted to see what your thoughts are on thinking about researchers, particularly pediatrics, and particularly pediatric infectious disease, as physician scientists, where we are, how do we demonstrate value to our institute where, in comparison to like full-time procedural specialists or, to a medical center, like how do you, demonstrate value do you recommend people demonstrate value and get them to kind of financially support you and support you through resources, especially in our fields that are well known as the low compensation fields, and then adding the research component on top of it?
Yeah. So that is like the $1 million question, maybe even more, maybe the $1 billion question to me, because I feel like I'm constantly trying to figure out how we can make research. Everyone knows it's important, but how can you quantify the value? Especially when the indirect costs of our grants don't even fund the whole research infrastructure that's needed to complete the research. And that's one lesson I had to learn. And I think something everybody does initially, like, oh, wow, I just got a $100,000 grant. That means my institution gets $70,000. Well, don't try to use that as a reason why they should invest in you, because they'll tell you, no, your research actually costs us $1.5 on every dollar you bring in. So you're actually losing money for us. So using your IDCs is not a productive way. However, what is productive, I think, is to say that you have funded your entire salary beyond what is your clinical effort. And for infectious disease physicians, because I know this world well, is you can't make enough work for a group of at least 4, right? You don't want people to be on more than 1 weekend a month. At least 4 members of an infectious disease team. A big place like Weill Cornell, UCSF, like you probably can't even fill 4 FTE of time with the work of an infectious disease pediatric, at least infectious disease physician. So therefore, you need researchers as part of that group. You in order to have a successful division like that, you have to have researchers, because you need more people to cover the clinical service and just spread the clinical work beyond. So that's one thing. And then the prestige, the reputation, buttons that I think about with when I'm trying to work with the health system is our U.S. News and World Report rankings is more than half reputation, and reputation is all basically through research presentations, funding, publications, where you quoted in the media. Like that's what, because remember, physicians vote for other physicians, or physicians vote for other institutions, and so that reputation-wise is an important one. But quantifying it, I still struggle with, even at the department chair level. So this is something I'm always asking people as well.
Thank you. It looks like Rachel Wadier has a question.
Yes.
Hi, thank you so much for this talk. I find it really helpful. You're so supportive and transparent, and I find your comments here like extremely balanced, and especially as a parent. And I'm not a physician scientist, so I do research, but mostly clinical and antimicrobial stewardship. But I don't want to derail too much, but I would suggest that the idea that ID physicians or other cognitive specialties can't be supported on their clinical work in a large medical center, I think is actually a myth. And I think in our profession, we haven't really taken advantage of all of the ways that we can account for the clinical labor. And so maybe just a plug to consider how we can account for the work both clinically and academically, because I think there are ways to explore, and it's just a matter of understanding the opportunities. But I think in our division, we actually are able to fully support the FTE and have actually been progressively hiring new people into clinical roles.
Yeah, that's excellent. And probably, and this is an area that I'm, and I appreciate your comment, that I'm working towards strongly now is support from the hospital. So, and every medical center is different in how they do this. And there are more and more metrics coming up of like, you've got to have antimicrobial stewardship and a certain proportion of FTE applied to it. You've got to have the infection control and enough FTE is supporting that. And luckily places like US News and World Report are pushing health systems to make sure that they have enough. And that's where specialties like ID, you can get more support like that. But we have to advocate for that. The health system is not generally like, oh, hey, here's some more money. No. So we do have to advocate for that. And, then what I will talk about in the last few slides is the importance of advocacy to change policies, to change how we can better appreciate researchers, as well as better appreciate those underpaid specialties. And in pediatrics, I'm constantly saying now, like, we have to fix the inequity of Medicaid reimbursement that is not near equal to even Medicare. So, and that is like the root of a lot of problems in pediatrics. So there is a role for advocacy that we need to apply and not accept not accept no for an answer. But as a department chair, I still say I need ID researchers, because otherwise I won't be able to cover all the weekends. So it is a work in progress, I would say. But thank you. That's the type of conversation that it's the type of experience I'm getting to continue to motivate me to fix this issue. Maybe I'll go on so that we have plenty of time for discussion, because I'm just going to run through a couple of vignettes on research. So I'm going to share again. Okay. All right, so a couple of vignettes on diversions in research, which I do think are important to take, also important to figure out when they're not working. But I'll tell you about some of the successful ones. So I initially aimed to study breast milk transmission of HIV, which now is responsible for almost half of the, really it's more like 160,000 infections that are occurring annually, but over half of them are breast milk transmission, where it used to be the minority of the transmissions before maternal antiretrovirals. Formula feeding leads to high rates of morbidity and mortality, often due to diarrheal diseases. ARV is effective in limiting transmission risk, but a lot of implementation issues there for keeping moms on it during the postpartum period. And then moms acquire HIV during the breastfeeding period, which is obviously longer than pregnancy. So, so it needs to be addressed, and is not being addressed by any of the antiretroviral strategies now. However, the thing that I initially started to study is why is it that even when a woman is newly infected while breastfeeding or on no antiretrovirals whatsoever, that only 10% of infants who are breastfeeding to an HIV infected mom will become infected? Like, why not 100%? What's protecting the other 90% and who remains uninfected? Really trying to understand who remains uninfected. So that was an initial area of pursuit. But one thing that was so annoying to me is that I was trying to study neutralizing antibodies in breast milk, and here's some uninfected milk, those first 10, and then some HIV infected milk. The uninfected milk always had high background. I would always get some level of neutralization across all the viruses, all the different variants, it wasn't, it was maybe 10-fold or 5-fold or so less than HIV infected milk, but it was still annoying that we had so much background. And then I started to think, well, what is that background? Like, why is it that just breast milk from uninfected women neutralizes HIV a little bit? And was talking to, a colleague, and they said, well, if you want to try to figure out what, set of proteins is doing that, we could do some fractionation studies and look at, which fractions are neutralizing and try to identify, where it might be coming from. So I said, okay, you have a size exclusion fractionation. I have HIV neutralization assay up and going, and I have the breast milk. Let's do this. So we took a neutralizing uninfected breast milk, did a size exclusion fractionation of the proteins, and by HPLC, and just remarkably, and I think this, often is not the case, but we had the neutralization activity was only in one peak, the peak 3. And then when you looked at peak 3 on the graph, it had like one band that wasn't in the other gels. So we did a proteomics on that and found this protein, tenacin C, which is the third most prevalent protein in breast milk. Its function had not been identified. It is found in extracellular matrix, joint fluid. It's turned on in development of the brain tissue. It's turned on and then turned off in fetal development, but then it sometimes gets turned back on during in tumors. But nothing, antimicrobial had ever been described of it. And this is its structure. It's a hexamer of these long domain arms that include EGF domains as well as fibronectin domains. And we then performed various experiments to show that tenascin when isolated, could neutralize HIV, and went on to map that it could be that its activity is through blocking the CCR5 co-receptor interaction with the CD4 molecule after its engagement, with the HIV envelope after its engagement with CD4. And that was like really exciting work. Ended up, it was one of these where I submitted it to I think Science and Nature, and like waited like by my email like every hour, like what's going to happen. I think it got reviewed at one of them. We ended up publishing it in PNAS, I think. But what was funny is one day my father called me and he said, "Sallie, you got to read this article in The Economist." He's like a big Economist reader. It's something that's like right up your alley about breast milk and protein in breast milk that may have been, protected babies all these years from getting so many infected by HIV. And I, opened up online or whatever, and I was like, "Dad, that article is about me. That's about my research." And it didn't have my name in it because it had the first author. I guess The Economist, focus on economy, how economists, write manuscripts. They focus on the first author and named the first author, who was my postdoc. But he didn't believe me. He's like, no, it's not about you. I was like, no, it is about me. So anyway, so that was a fun like little first project. The next diversion that I'll talk about is like really a motivation that came from my clinical work, where when I got into my pediatric infectious disease fellowship, and then as a profession, I started seeing how many kids that we were seeing, and we had a congenital infection clinic at Boston Children's, that almost all of them had congenital CMV. That was the most common infection we were seeing. And then, reading about it, it's happens in every 1 out of every 150 to maybe 200 babies throughout the whole world. And that just in the US, that's 40,000 cases annually, 5,000 go on to have permanent sequelae. And that's the most common cause of long-term disabilities in babies. It's the cause of 25% of all infant hearing loss. And for all these reasons, it's been a tier 1 priority vaccine for now more than 20 years by the Institute of Medicine, or it's actually National Institute or National Academies of Sciences. But we don't have a vaccine yet. And I knew a lot of HIV vaccine researchers. I didn't know any cytomegalovirus vaccine researchers. So I thought, maybe I should try to study this. So my thought of how to study it was, could I apply non-human primate models that I was using in HIV work? I'm sorry, these are on a timer. Sorry. One more. Sorry, my slides are ahead of me. Okay, could we create a model of congenital cytomegalovirus in non-human primates? It's really hard to do vaccine development and preclinical development because the CMV viruses are so species specific, and so you can't challenge a monkey with a human virus or a guinea pig or a mouse even. So you can't really do preclinical challenge models. So then how CMV vaccine development has been really delayed is you have to do a big human trial in order to figure out if it works or not. So I thought, well, if you could develop a non-human primate model, maybe that would be a better preclinical test before you planned all those clinical studies. And so far, the fetal infection of rhesus CMV had occurred and shown that could develop a human-like disease, but no one had infected pregnant dams who were seronegative and determined if it could cross the placenta. So we did that, but like, how do you even start a study like this? Because like no way an NIH grant is going to fund that. And so I applied for the DP2 grant that, sorry, I'm having a hard time with the timer, that proposed this basically, that we would, I'm going to stop messing with it. We would infect seronegative monkeys that have been developed at certain primate centers, mostly to get rid of the herpes B virus of rhesus monkeys that is infectious to humans. And so there was an attempt to breed out that virus by taking herpes virus negative monkeys, by raising them, harvesting them early from their moms, and then raising them in a herpes virus free environment. So we made use of those animals. We infected a first few dams that were seronegative and saw if we could see transmission. And the data is there on the top row, that we did have some evidence of transmission looking at amniotic fluid in the blue there after a primary infection in the dam. But I was a little underimpressed that we couldn't find any disease in the infants when they were born. Not even liver enzyme elevations, or we couldn't find virus in their blood. You couldn't get urine right away, but so we maybe we looked for it in saliva. So I wasn't really impressed. So I thought, okay, can we immune suppress the moms and see what happens then? And so we did a CD4 T cell depletion, which a colleague of mine had shown does increase the time period of viremia. And there we saw 100% transmission and high rates of fetal loss. So then I thought, okay, now we have a model that we can use. But basically proved that yes, the non-human primate version of cytomegalovirus can cross the placenta, unlike a mouse cytomegalovirus cannot. So this ran in, allowed for a new model. Here was just confirmation that we found the virus both in the placenta, fetal tissue, and by PCR and immunohistochemistry. Since this diversion and writing this grant, that when I got it, I thought like, oh my gosh, no, I actually have to do that work. It seems like science fiction now that looking back on what I wrote, but it has led to a program project grant, several R01. So because of taking that diversion and studying something that, or writing up a grant that really I had no business writing at the time, has developed into a whole area that we've now been able to capitalize for advancing the CMV vaccines. So another diversion is one thing that I've been interested in is how we can take advantage of the natural pathways that in the maternal fetal infant immune space is the passive transfer of antibodies. How, now that we're in the antibody evolution, biologics, antibody engineering, how can we take advantage of that to better protect infants in those early months of life? And I'd studied a lot of breast milk, and so thinking about could dimeric IgA be an engineered antibody that would provide extra protection to the breast milk compartment that then would be delivered to the infant gut. This really came as part of a diversion because I was working with or had my office next to really a PI who led the non-human primate reagent program, where they were developing rhesus antibodies, and he had made a rhesus dimeric IgA and a rhesus monomeric IgA. And he said, I really want to test this functionally somehow. Would you have anything you would want to do with these antibodies? And I said, well, I have a lactation model in non-human primates. We could see how well they get into breast milk. He's like, great. So somehow, I don't know, we funded a couple monkeys. This was like 4 monkeys. And we infused them with either IgG, monomeric IgA, or dimeric IgA while they were lactating. And we did find that the dimeric IgA, first people told me, this was another, people tell you no, don't believe them. Oh, there's no way a dimeric IgA will get out of the capillaries and get to the poly IgG receptor and then get transferred into breast milk. And we did find the antibody in breast milk, and it was actually the most persistent, and therefore we got the most neutralizing activity out of this. This is a neutralizing activity again, a neutralizing antibody against HIV. So then that led to work that we're continuing to pursue now, which is could you engineer a dimeric IgA antibody to administer to a woman in the postpartum period, for the goal of making her breast milk more protective against enteric diseases like rotavirus. And so this is some work of my postdoc now assistant professor, Stephanie Langle, and my collaborator, Maria Blasi, where we produced a known neutralizing antibody against rotavirus as a dimeric IgA. They're shown in the EM pictures, and showed that we can get it into breast milk of mice. Now, this was a real challenge going from non-human primates to mice, but the Gates Foundation made us do this because they were giving us a grant and they said, we don't want to do monkeys, it's too expensive. And I was like, well, I only do do monkeys. And then I think it was Stephanie who convinced me, like, no, I study mice too. Let's see if it happens in another species. Because we were like, probably the dimeric IgA won't get to the breast milk in mice because we only saw it in primates, and I don't know what happens in mice. And she said, no, let's try it. And there we did find that infusion of the dimeric IgA did get into breast milk. And we could even find it in the pup stomach contents, and then went on to show that we can reduce rotavirus-induced diarrhea with this strategy in pups born to dams that were given the dimeric IgA that has a neutralizing activity against rotavirus. And last aversion is, of course, pandemics. So if you're a virologist, especially if you're a virologist, when everything else is shutting down except for coronavirus research, you pick up coronavirus. And I think, as pediatricians, we, first of all, we're amazed, like, really, infants are not going to be severely affected by this. They truly weren't. It was really a respiratory virus that, for whatever reasons, which I think has to do with interferon, I think we've come to, affected the elderly and adults more severely. But of course, children were not unaffected. And I had this feeling going in, like, definitely kids are going to be left out of this vaccine pursuit. And I didn't really know what to do other than, like, let's study it in a non-human primate model. So that's what we did. We were able to capture some of that, amounts of money that were being added on to existing grants in order to study this. I'd already worked with Moderna because they were working on a CMV mRNA vaccine at the time. So called up Moderna, like, could we get some of your vaccine in order to give to infant monkeys? I know you're already doing adult monkeys. And then also collaborators at the Vaccine Research Center who were making the stabilized spike protein. And can we compare a protein in an mRNA vaccine in a non-human primate model of infant vaccination that we were already using for HIV vaccination? And so we did that, compared an mRNA and a protein vaccine. Definitely the protein vaccine elicited higher magnitude neutralizing responses. We are using a very potent adjuvant against TLR7/8, the 3MO52 adjuvant, which we already knew was a potent adjuvant in the infant immune system. Higher magnitude neutralizing antibody responses, but durable responses in both. These were 2-month-old infant monkeys. We then when Delta was king, we chose the Delta virus to challenge the monkeys. And we challenged them a year later after only 2 doses, and established that really only the protein vaccine could knock down replication of the virus. This is a heterologous virus, since the original, the vaccines were developed on the original virus. But only the protein vaccine saw reduced virus in both nasal swabs and lung samples. But both vaccines definitely protected against lung disease. However, there were a couple of animals in the mRNA group where the neutralizing antibodies had waned and did have some lung pathology that really demonstrated here that those 2 yellow triangles and diamonds shows that those animals that had a lower neutralizing antibody titer were the ones that had some lung pathology that was similar to that of controls. So now, with all those diversions, this is kind of the slide I put together with what my lab does. And really, to me, is an exciting portfolio that then I'm able to plug mentees into in various ways. Are they interested in HIV? It's funny that just like the NIH, many trainees come to me and they're either interested in HIV or other viruses. And so I still am able to sort of plug people in that way. And also, with having a diversified portfolio, have also had no problem with people that come into my group as postdocs and then are moving on to assistant professor positions, taking some of it with them, because I cannot do all of this myself. so that has been an advantage as well. So let me just check the time. So I just wanted to talk a little bit about what do you do in your mid-career, in your senior career? You've made it as a physician scientist, and now how can you make the pathway easier and more fertile for the next group? And so things that I think about is mentor at all levels, and think about down to those levels where an early experience can just change a career completely. And so I try to keep a diverse group, and even do what I can at the high school level especially with our department and clinical experiences, volunteer to organize trainees. There's never too much networking. Time is always an issue, but and this is the way I think I really started to just see that was necessary, and then became known for being someone who was good at organizing mentors and mentoring groups, and then became the assistant dean of physician scientist development through that pathway. Write a training grant. So I remember at one point in my career a few years ago, I thought, I really need to know how to write a training grant. So I just volunteered. To my department chair. There was a training grant that had been submitted and didn't get through. And I said, well, can I try to rewrite it and get it through? Yep. So that was a great experience. Support diversity at every level. Like that is something we have to get better at, and get rid of the barriers that exist. And again, early experiences, and looking out, being that ally, and doing extra that you need to do in order to take those diverse candidates through to the highest levels in their field. Advocate for training opportunities, funding, research in your area for your patient population. Also health equity. I think health equity is a component of every single disease we study, no matter what field you're coming from. And I hope that someday the NIH not only will ask us for sex as a biological variable, but health equity and how the work that we're doing is affecting health equity, even at the basic science level. And so that is something I think we could all start thinking about now, because how we're delivering healthcare now is a shame, how disparate outcomes are. I advocate now. CMV has been a great opportunity to advocate because it's a virus that has a PR problem. It causes a lot of disease, yet few people know about it. We need to tell more people about it. We need to test every baby, and I'm doing a lot of advocacy there. We just had a law that was signed into a bill or a bill signed into law last month for expanding education for CMV in New York State. Next, we're going for testing all babies, which is happening in some states. I also work on the National CMV Foundation board. This is my mentee, Claire, who was just an awardee for research dollars that they did not really give out research dollars until I got on the board. And I was able to kind of advocate for the importance of research and early career research to get someone into the field. I also communicate to the public. I did a lot during COVID because I knew kids were going to be left out. You could just feel it. And so I continued to advocate at that level. And now I have a policy grant, which is like a whole nother world, on how we can design models for the FDA and companies to incentivize vulnerable populations being included in research, including pregnancy and children. Also, we've been experiencing a respiratory virus surge in pediatrics that really didn't get the attention that it needed. I just wrote an op-ed on that. Yesterday, I was spent the day at the governor's office giving a press conference with her on RSV. I think despite the fact that I wrote an op-ed that said the president should call a national emergency, they vetted me a little harder because they read that op-ed, because they didn't want me to say that on the press conference. But this is an important way that I think we all know how important it is to communicate our science. So many people to thank. I had a ton of trainees that helped me get me to where I am, but I'm going to stop and use the rest of the time for any questions. And I see there's some in the chat.
Thank you.